Well, thank you, everyone. Welcome to ASBX 2021. And this afternoon, this live discussion panel is about the translational research and of microgravity research to terrestrial settings. I'd like to thank everyone for joining us here today, and I'm truly honored to be able to invite our panelists who are all respected world leaders in what they do. So it's a really great privilege to have them to join this platform in which we can discuss a lot of the achievements and challenges of facing translational research and microgravity research. So I would like to quickly introduce all our panelists, and afterwards they will give you a more in-depth introduction about what is it that they do, because their accomplishment is very long. It'll take me a while to, and I probably won't do them justice by introducing them. So today we're joined by Dr. Giulia Silvani, she's a postdoctoral research fellow from the University of Technology, Sydney. And we have Professor Volker Hessel joining us here today. He has a lot of experiences, so we'd love to hear about all his research and projects that he has going on. And we have Melanie joining us from ANSTO as well, and also Siddharth Pandey joining us here today. So I would like to open the session, and I'll let Giulia Silvani to introduce herself first.
Hello everyone. Hello Joshua. Thank you for the introduction. And yes, I'm a biomedical engineer, a mechanobiologist, and now a space cancer researcher here at the University of Technology, Sydney. And my background is very multidisciplinary, spacing from biomaterials, biophysics, cell biology, and mechanics. And I work with novel strategies and technologies to develop increase the knowledge and understanding of cancer disease. So sometimes I think that I've been really lucky because I'm working in the most beautiful place in the world and on the most exciting field of investigation, which is space biology. But honestly, I don't think it's a matter of luck, but I think I have a very strong passion for science and an unlimited level of curiosity. And so when I graduated at the University of Sapienza in Rome, when I come from, in bio-nanotechnology, I was basically searching a way to keep studying and feed my curiosity. And that sounded like a PhD, which I completed at the Italian Institute of Technology in microfluidic applied to biology. And I guess this was true. Like I keep studying a lot and more than expected, but those 3 years have been the most amazing of my life because I gain so much knowledge about interesting subject, and I become an expert on sophisticated technology. But most important, I connect with brilliant minds that have been inspired me even more to pursue this academic career. And one of those was Boris Martinak from the Victor Cheng Institute here in Sydney. So what really happened is that I had probably the courage to propose myself and my contribution for the Martinak team. And as soon as he agreed, I was already flying on a plane over the Pacific Ocean, dreaming about this beautiful life made of sun creams and science book on the tropical beach. But honestly, my fantasy went too fast again because it ended up being a very tough year for everybody because of the global pandemic. And so I was working really hard to keep this dream alive. And I remember I was working on this sophisticated spectroscopy tool called the acoustic force spectroscopy when actually you came into the room with your enthusiasm, introducing yourself as a space biologist and interested in collaborating with the team. And that was great, a very big opportunity for me, not only because I've always been interested in the mystery of space, but also because I think that sometimes it's really important to change perspective and point of view in order to allow a more critical and fresh mind for the interpretation of scientific problems. And what I'm doing now is looking at the sky, searching for answers from the space environment in order to advance our knowledge on cancer disease and facilitate the development of new treatment actually to fight this deadly disease. And in particular, I'm interested in glioblastoma, which is the most aggressive brain tumor. And I'm working at the interface between space mechanobiology and novel 3D bioprinted model, which can allow to better mimic the microenvironment of the tumor and investigate the impact that microgravity can have on tumor cell function and dysfunction. So far it has been an amazing journey, and I guess the best is yet to come.
Thank you so much, Julia, for your introduction. I think she really sets the stage. It's about diversity and different perspective, and I think sets the foundation for today's panel. Is that all our panelists come from a different diverse range of backgrounds, but with the common theme of translational research and how we can all be part of that, and as well as our attendees. Not only understanding what is it that you do, but more importantly, how they can contribute to the overall space ecosystem. So thank you, Julia. I'll ask Melanie next to introduce herself to the audience, please. Thank you.
Thanks, Josh. So my name is Melanie Faletza. I have a PhD in radiobiology, and I'm currently a postdoc at ANSTO. Arrived a bit more than 2 years ago at ANSTO in a collaborative project between my former lab, which is the unit 1296 INSERM in Lyon. It's a radiobiology unit. I think it's the biggest in France at the moment. The French Space Agency, where I spent 2 years as a postdoc before arriving in Australia, and of course ANSTO. The idea with that project here at ANSTO is using the Center for Accelerating Science to produce secondary radiation that are similar to what happened when cosmic radiation interact with shielding. And the idea is to study this particular radiation on human cells. So that's what I've been doing. My background is DNA repair. That was the main subject of my PhD studies. So we've been developing that with Center for Accelerating Science at ANSTO, and also during the past 2 years we made connection with Joshua's team, and we have a project to study the effect of microgravity combined to radiation to try to mimic as close as possible what really happens in space. So yeah, well, between COVID and everything that happened at the moment, we try to work on review work and everything, and yeah, everyone is in the same position at the moment. But yeah, the idea is to look at DNA repair after radiation to try to either give alternative shielding or propose countermeasures such as drugs to improve DNA repair.
Thank you, Melanie. And yeah, it's been really interesting working with Melanie and her group, and I think what they're doing is very important. Is we all know radiation plays a significant role in biology, but we don't know what are the mechanisms behind it, and more importantly, what we're trying to do is seeing whether it's microgravity or is it radiation or both that is having the effect. And I think that even though it's considered, I guess, some form of basic science, it ultimately will lead to developing better countermeasures or understanding of the human physiology exposed to these extreme environment, which are all relevant here on Earth. So thank you, Melanie. I'd like to ask Siddharth to give an introduction about himself. Thank you.
Thanks, Josh. So hi, everyone. I'm Siddharth. So my background is actually from engineering. I've entered the space ecosystem from an aerospace engineering background. I'm currently in Mumbai here in India. And that's actually where I was born and also started my journey. My education took me first to the Netherlands outside of India for a master's, and then from there moved to the US. I was at NASA Ames Research Center for a couple of years as a student researcher in the capacity of hardware engineer. And that actually gave me a chance to work closely with space biologists, and that was kind of my foray into space biology. I'm not sure if someone's screen sharing. Okay, I'll just keep going. So basically, my first experience was developing small compact hardware for observing platforms on the International Space Station, and I was working closely with the Fruit Fly lab there, and that's where I really understood the challenges that are associated with handling, live payloads and what kind of challenges that they bring with them, and that's something that we weren't really exposed to as engineers. So working with life science teams, that was kind of my foray. I decided to continue getting my education, moved to Australia, and that's actually my Australia connection. I was at University of New South Wales, Canberra for 4 years, working on a different challenge, thermal modeling for Mars rover missions. But the interest to kind of help support and grow space biology and also a bit of astrobiology, that's another thing that I was exposed to while I was at NASA Ames, kind of kept going. And I moved to India about 3 years back. And since then, I've been helping set up a research center here, which is the first dedicated center that we have in India for astrobiology. And we include space biology within astrobiology, and we work closely with several national and international partners. So there's primarily 3 things that we're doing, but I just, for the paucity of time, just focus on the space biology part of things. So we've currently been selected to fly 2 experiments on the Indian Space Research Organization's PSLV mission, hopefully next year, assuming things go fine. And my kind of establishment of this center was from my own personal experiences and background of technology development. So we were really focusing on building modular, light, compact, non-recoverable payloads that can essentially support experiments that require growth observation or any exposure to radiation. As of now, we're mainly focusing on growth observation, internal payloads, because the platform that we currently have from India is not allowing us to get the experiments back. That's one of the limitations. But so whatever we can do within limited bandwidth to get the data back from your payload, and also be able to also handle temperature and pressure, extremes of space. So that's one thing we're doing. And currently we're working with our own in-house biotechnology team, and the samples that we're flying are plant callus samples and bacterial samples. So the team is essentially looking at high-yielding food crop materials that would be useful for long duration, self-sustaining, crops that we can grow in space. And from the bacteria side, we're looking at probiotics. So there's several probiotic samples that have been isolated for the Indian gut digestive system. So essentially right now, our science is to support the upcoming Indian human spaceflight program, because that is currently our use case, because we don't really have an Indian market for microgravity research as of now, at least. But we are very encouraged with how things are developing here in India, and there is a growing ecosystem of new space startups, as well as entry for foreign researchers to come in and use Indian microgravity opportunities. So with all of those kind of things, we're kind of growing strategically. We're not trying to do too many things. We're just trying to make our own little niche that will help us stay relevant and help us build collaborations. And yeah, that's my side of the story.
Thank you, Siddharth. And I think you've touched on a lot of key points. It's about, it's an emerging market, and I love how you're from an engineering perspective, but looking at, life payload, because I think that is really challenging, because sending electronics is, relatively straightforward, because, it's not living, but sending live samples to space or ISS is, is really, really challenging. So I think that will be also one of the focus of discussion today is, where are we in terms of the technology, and where would we like to be in a couple of years' time to support or expand or advance biological payload. So no, thank you so much. I'd like to ask Volker to introduce himself, and we'll hopefully see the relevance of all our panelists' expertise in today's discussion.
Yeah, thank you so much, Josh, and all the organizers for this great panel and the opportunity. I'm Volker Hessel from the University of Adelaide, and indeed, space research is very translational, and my retail is a bit aligned on that. I'm a chemist by education, but what I basically do is that I create and develop new processes, chemical processes mostly, and we did so here at Microfluidics. We started with microreactors almost 30 years ago, and a lot of those microreactors are hanging there, which my colleagues gave me when I left Germany. There's quite some value, you can be sure. And microreactors are disruptive technology. And what we basically made was a mission impossible to convince pharmaceutical industry to make their chemistry through something which is not bigger than my hair. My hair is 80 micrometers. And finally, they do now the COVID things, and all things are done in flow chemistry, continuous flow. So we made that happen. That industry took it, and I thought space, that is the next big water. That's even more impossible. So let's do that. And I'm just happy, just today, I think the Australian priorities were announced, and flow chemistry has been set as a national priority. I was completely surprised by this because it was not a big topic before. And it fits quite well that together with ISIO, I brought the first international conference on microreactors to Australia, which happens here in December in Melbourne. Now, what I do in space is quite diverse because the technology can be applied to many things. I have 8 PhDs in space food, space architect, and space mining and space medicine. And I'm part of a Center of Excellence plants in space, which has applied here where good Australian plant researchers and bio researchers have aligned. And I run the, to my best knowledge, first Australian space experiment at the ISS. And these are the tablets that just came back this year. Probably some radiation, so better take them away. And that is the mission badge with the University of Adelaide mentioned here. I see the Frontiers logo in the back. Just since yesterday, I know I'm editor-in-chief of a new journal in Frontiers, which is space exploration. So the human space exploration is very important. I also make a book on space resources and space manufacturing. And I like to end mentioning our nice center. We have the Andy Thomas, and I think you all know who Andy Thomas is. The Andy Thomas Center of Space Resources. Andy Thomas comes from North Adelaide, just 3 kilometers from where I sit now. And he studied in our university, in our faculty. And the Andy Thomas Center students have made the first place of all universities in the NASA Robotics Challenge. From 114 teams, they were the first university worldwide, so we are quite proud of this. And we built a 3 square kilometers wide Mars surface at the Roseworthy campus. Yeah, 3 square kilometers, that is big. To my knowledge, bigger than what ESA has. And we have here quite interdisciplinary teams, psychologues, architects, plant biologists, we all work together. So we make a microcosmos of this great macrocosmos, which I see here. Thank you.
Thank you, Volker, for the introduction. As everyone can see, Volker has a wealth of experience, and I always he's my go-to guy, anything to do with space medicine, pharmaceutical, and chemistry, because my chemistry is not that great. So it's good to have someone that is knowledgeable in this area. Okay, thank you everyone for your introduction. As everyone can see, all our panelists comes from a very different perspective, but nevertheless, it's all both center themed around not humans in space, but also that translational research of the processes or the technology back down here on Earth. So I would like to start off the panel, today's discussion, with the first question for all our panelists. It's a really, just to get the conversation going, how do you see the future of, whether it be biological or pharmaceutical research in space? And what do you foresee as some of the challenges? So what I'm really asking here is, where do you see the potentials, and why should people, should be interested in this area? So if you can give us, your glimpse of, why you're in this research and what are some of the values of this research to space and on Earth, that would be great. So we'll start off with Julia, and we'll just go down the panelists.
Yeah, so, well, I guess there is a lot to do, definitely. And what can I say is just that there is nothing impossible in science, especially looking at the space. It's so all unknown at the moment, but we have already demonstrated that the human can actually go very far, even with the mind and the body. For example, until last year, I was very skeptical about tourism in space. And just 2 months ago, the first all-civilian space flight was orbiting around the Earth. And that's such a great achievement. And Of course, there are several limitations. For example, not all researchers can just buy a ticket and go doing experiment in space, and also all the hazard and risk that we are exposed out there. But I guess it's a matter of time, and it comes with the technology development. But I can say that definitely we need to trust in this new field of investigation, which can actually really have a significant impact also to improve the quality of life here on Earth with translational medicine and all the discoveries that can be made out there. So definitely I can see the future for space biology, and I look forward for it.
Thank you, Julia. And you're absolutely right. I think the key word is, there are endless opportunities in this area, and how do we actually, how can people get involved in it? And there's so many discoveries to be made, especially as you said, this year we've seen so many activities of, and we'll definitely see a lot more going into next year of humans being in space. But are we actually ready yet?
Yeah, actually, if I can just add that, like over 50 years of human activity in space already have bring so much social and technological benefits. So I don't see why space biology can do the same. It's just, as I said, a matter of historical events coming with the technological and social also development. So again, we will see.
Thank you. And over to you, Melanie. What do you think about some of the challenges and
I think I'm gonna go the same way as Julia. Is space is such a constrained environment that all the technology that is needed to go there will be helpful here on Earth to facilitate a lot of process. If we talk about space radiation, radiobiology, at the moment we're using what we know from clinical radiobiology, so cancer treatment, radiation therapy, to try to apply those models and the biological pathway that we know are activated after radiation to space to have an idea what happens when human is exposed to this type of radiation that we find in space. But I think all the development that will go with countermeasures will also be helpful on Earth to protect the patient from radiation therapy treatment after effects. So those skin burns for people that will have bad reaction to radiation therapy, for example, or also improve radio protection for people that works in contact with radiation. So, and I think it's a valid point for every part of biology. We're using what we know at the moment for the specific pathway that we study, and then with the new data, we just go back to what we know from Earth.
Just to follow up with you on that, have you seen any similarity between the radiation exposure here on Earth and in space, or is there differences between the two?
So for space, the main problem with radiation is that it's a very mixed field, and it's hard to recreate the exact same one. So what we do at the moment is that we do separate types of radiation what we can find on Earth with the technique that we have on Earth. So yeah, well, radiation breaks DNA, and you can see that. The improvement would be to have, if you don't want to go to space, is to have like a mixed field that would be as close as possible at what we find.
Okay, no, excellent. No, I think it's a very interesting question. Radiation is indeed an issue, not here on Earth, but also in space as well. So there's definitely a lot of cross-translation.
And maybe that mixed field will be very efficient to cure cancer, for instance. I mean, I'm just thinking out loud, but that could be one of the way to go back to Earth.
Exactly. No, thank you. Siddharth, you come from a very different perspective, but from the engineering perspective, what do you see are the opportunity? Because you're obviously working developing these technology, but there must be some value of it that makes you being involved in this area.
Right. So definitely, I think I'll agree with Julia and Mel on this. So technology is improving. It's always at a constant stage of improvement. So what the kind of things we're able to do today is obviously a lot more well equipped as compared to what was possible, say, even in the early 90s. And we see that with the kind of hardware, whether that is for microfluidics or any sensor driven things, lab on chip, tissue on chip kind of things. Definitely there is an improvement. in terms of access to space, also things are improving. We will probably see a lot more privatization of space, in the sense that there will be access to lab spaces probably in the near future. Aside from what we have on the International Space Station, there might be other private players who will be launching up platforms that can actually be accessed. So all of that is really good. I think, so in terms of building the use case, because there's obviously a lot that can be done, right? We're talking about so many different kinds of sciences and so many different kinds of very critical areas, whether that's from any part of the body. There's so many things that can actually be done, but the limitations come with what is possible currently with automated systems, with things that are kind of self-equipped, right? Because astronaut time is very, very limited. So we will reach a point where there's only so much that they're able to do that is sufficient for that science to grow. Like they might be able to demonstrate a certain piece of technology that's kind of frontier in its end, but to take it from point A to point B and point C, it requires them to spend more time, elaborate amount of time to conduct those experiments in space. And there might be a small lag there, because we might not see teams of medical researchers flying into space to do that kind of stuff. So that might just be the only reason that could be probably holding back. At the same time, all of these facilities that we have here on Earth, these radiation facilities and other simulation facilities, are also expensive to maintain, and there's only a limited number of researchers who use it. So there's definitely a case for us to send samples up into space and carry out whatever experiments are possible given the space environment. So it'll be interesting how the next, I think, 5 to 6 years will play out. I think dependency on the International Space Station will definitely reduce. So we'll definitely have more free-flying experiments, as well as other kind of platforms where we can do stuff. And countries like emerging countries as well, where there is like already a lot of medical research happening, they will have access to flying experiments into microgravity as well. So I think the only challenge really will be the number of humans that can actually go there and kind of assist the experiments from taking place.
No, thank you so much, Siddharth. And yeah, you're absolutely right in that a lot of the, I guess, the biological payloads right now is it has to be automated, because astronaut time is very limited and also very costly. So I think it also limits the type of experiments that can be done on the International Space Station. So hopefully, as we develop the space economy in space, things will improve and we can expand the type of research we can do. No, thank you so much for that. And over to Volker. You have extensive experience and vested interest in different areas, resources and pharmaceutics. What do you see as the next? What are some of the value and what are some of the challenges?
Yes, I like to go on what Siddharth said. I see space, first of all, as a laboratorium, and as a perfect laboratorium, and seeing the increasing number of space papers, the question once really has to be, do the experiment on Earth or do it on space? Yeah, we have the same batch or flow, and the answer is always batch and flow, and so the answer will be to do experiment on Earth and on space, where it's best, and I comment in a second where I think it's best. We have something in between, fortunately, that are the simulated space experiments. I know, Yoshi, you built random positioning machine. We have here one, so we can simulate microgravity. We can simulate the space radiation in a reasonable manner, so we can bring the space experiments to us if we really need that. So we have 3 opportunities. And what I see really as breakthroughs from the space research, as far as I know them, with regard to biology, it's the crystals. Crystals form very slow if you want to have perfect crystals. And it's a very complex process, which not so many people here on Earth can do. In space, we have a bit more better conditions. So we know that better crystals form both from the antibodies, meaning the disease side, but also from the drug side. It has been shown. A good colleague, or could even say friend of mine, has brought the COVID drug to space. Yeah, the Hungarian mission, that's the first COVID experiment done on the International Space Station around about half a year ago. And also tissue engineering and body on a ship and organ on a ship works much better in space, basically because gravity compresses cells on Earth. cells like that, and we have much better physiological conditions in space. So it's a more perfect laboratory, but it has also naturally disadvantages. And finally, I like to mention that there are big unsolved problems from space, biology problems. We all believe that chirality comes from space, that the amino acids come from space, or the origin of life. And there's a famous Yuri Miller experiment from 52. This becomes 70 years old next year. And this experiment is still not answered, and people are still investigating the original bottles from Urey and Miller. I think they were in Chicago. So that is still a hot topic. Where do the amino acids come from? And you know, without amino acids, no humans. So that are interesting questions to solve.
No, thank you, Volker. And since you're already on the topic, I would like to follow up the next question with you. There's obviously with more humans in space, they're going to be facing a lot of different conditions. I want to discuss with you about space pharmaceutics, about formulation. I think a lot of people are very curious about how drugs or medicine works in space, and of course exposure to radiation and microgravity will affect its efficacy. Do you have anything to can you share with us what your thoughts are on that?
Yeah, I think in the last 10 years, to my best knowledge, since 2012 roundabout, we have quite extended studies on drugs in space on ISS, roundabout 6 or 7 missions, if I'm not mistaken. And one of the most famous one is from Watring, and I know that was a speaker at your conference, and they studied the whole ISS medical kit, so 35 drugs, and roundabout 50 other drugs have been studied, and it shows a mixed picture. Some of them, say half of them, decay slower or faster. Some are not impacted, and but the fact that 50% decay and decay faster usually than on Earth makes us thinking. And then we come to formulations. Do we have to have a liquid formulation, like the COVID, or do we have a solid formulation, which is a tablet or capsule? And what is actually better, a tablet or capsule? In space, we learn basically everything on our own. And what we tried in our experiments was to make a cosmic ray shielding coating to apply it. And the first experiment shows it might have worked. We have to investigate further, but we need to learn a lot about drugs in space. When we go to Mars and back, it's 2.5 years, assuming you stay for half a year there. No drug on Earth, or hardly any, has a sheet life longer than 1 year, and in space they're shorter. So we cannot really, in my belief, let people go with drugs which are 3 years old. Yeah, and that under these harsh cosmic ray conditions.
No, that's very interesting, and I think that again, what you're identifying is a lot of opportunities, and also challenges we have yet to address yet before we really go into deep space or prolonged periods of time. No, I think both Volker and Siddharth have already gave us a teaser about simulated microgravity, and that's something that obviously we use as well. But I'd just like to get everyone's thought on simulated microgravity, because right now I think more traditionally people just take an idea or concept and then go straight to the International Space Station. Obviously that's very time consuming and also in some respect costly as well. So I'd just like to get everyone's thought on where do you see simulated microgravity fit into your particular area of research? And simulated microgravity has been around for quite a while. So why hasn't more people used it? And given now that there's more interest in biology, health, pharmaceutics, do you see more potential, more applications of simulated microgravity playing a role in that whole process of between lab, potentially simulated microgravity, and then to space? So maybe I'll start off with Julia on getting your thoughts on where you see simulated microgravity.
Yeah, I've been working this year on simulated microgravity, so I really believe that the value of simulated microgravity is huge in this race toward the space discovery and everything. And this, especially in my field, which is space mechanobiology, because microgravity, simulated microgravity in this sense relies on devices, on machine, like the random positioning machine, the one that we have in the laboratory. And so these devices are able to really closely mimic the mechanical environment on space. And going beyond all the limitation, economical limitation or safety limitation, and this is a very important alternative for researchers that can actually get into space for make their research, and this can really help to advance and accelerate the process of discovery. But of course, like as Mel said before, it's a compromise. You can't really have the exact environment in space on Earth, but definitely a little step can be simulate as close as we can the environment and do the experimentation here, while the technology can develop and allow us to get to the real space environment. And also with the random positioning machine, we have seen there are several limitations. So still a lot needs to be done here on Earth, but I would say that it's a very good start point to see other progress on this.
No, thank you so much, Julia. And yes, I totally agree with you. You know, biology is already complicated here on Earth, let alone trying to do in space. So to have that is actually of critical value. I'd like to ask Melanie to follow up on that, because Melanie was mentioning before, even though radiation, they can't do 100% mimic of cosmic radiation, but we can do selective or broad range of radiation that's simulated. So where do you see simulated radiation and simulated microgravity, and what do you say to those people who say, well, this is not really the real thing? Where's that where do you see that translation and value?
Well, this is definitely a step closer to the real thing. So that can allow us to do not mixed field, but do different types of radiation within that microgravity environment, and then have information how the DNA repair will react. Also, it's an extra stress, because as you might know, different stress will trigger DNA repair mechanism, and it's also a way to study just the biology of that type of stress, and have idea of what protein are activated specifically, and if DNA is a bit not happy in that condition, so we can have a look of the biological process that are involved in just the microgravity alone. But of course, the idea of having the radiation plus the microgravity, it's one step closer to actually sending cells into ISS or into in space.
No, thank you, Melanie. And I think you touched on a really good point, that it is a stress, a stressor for the cell, and that will induce a reaction, response, and that's what we ultimately, as biologists, is really after, is how do they respond to that? It doesn't necessarily have to be 100% space environment, but it's a different environment. It's an extreme environment in itself. So thank you for that. Over to you, Siddharth. Where do you see simulated, I guess, versions of microgravity or simulators, and do you see it playing a role in that whole space biology or process between Earth and space?
Yeah, so I think to start with, especially from my perspective, when I was in the US, definitely we had access to a lot of facilities, and moving to India, there was a lot of shortcomings when it came to getting access to facilities. Those things are changing. I think there were just 1 or 2 companies that would provide RPMs and clinostats, but things are definitely changing. My students actually had been trying to develop their own systems, just 3D printing some parts and using motors and finding ways in which you could kind of rotate them at different frequencies and recreate similar kind of rotations. And one thing I came to know was that definitely it is the first step. In my opinion, every group should definitely explore and understand the effects on Earth, and try to rule out the other stresses first, and then from there kind of build on, and then do the experiments in a reduced gravity environment in actual space. There probably would be some kind of a ceiling that it kind of gets hit, because of you're not able to completely replicate the reduced gravity environment in all the 3 different axes, right? And it again depends on what kind of experiment is being conducted. Some experiments might not be so sensitive to gravity, and you might be able to get a lot of useful results. Whereas there might be others that are working at a much more smaller molecular level where, you can do like your initial test there, but you actually need to, fly into space. That's kind of been my experience. And I think also the form factor of the experiments is a large deal as well. Like for smaller experiments, you can definitely use the conventional ones, but if you're trying to do something that's slightly larger that might not fit in the existing platform, that also becomes kind of an issue.
No, thank you so much for your perspective, and I totally agree with you. I think it has a role, and like anything else, like everything else, it has a role that it plays in the whole process. But of course, it can't cater for everything. So no, thank you for your your thoughts on that. And Volker, I know you've worked in both simulated and also in real space. What's your experience in using technology?
I think the right keyword was given, that is stress testing in those simulated environments. And we have the unique opportunity to test, for example, when you think about plants and crops where we are busy, what do we actually take up there? Tomatoes or pepper, or what is more resistant to it? And the likely answer is also that I'm not a plant biologist, but that stands to reason that we have to modify those plants to be more resistant. We can do that all on Earth and avoid failures in space. I think somebody also wrote that in the online chat, that this is a great opportunity. And then I always say stress is not bad. Stress makes us great. Yeah, there's use stress, and so you can positively use stress to do something on Earth which otherwise might not be possible. And finally, Josh, I like to understand more. I need to read more literature about these random positioning machines and the microgravity, what it really is. When I want to make a crystal, I don't want to have any convection. Yeah, convection is anything which goes around on a larger scale, such kind of eddies. But when I see my random position machine mixing my fluid, it's constantly mixing. So I'm not so sure what I can take for granted from those experiments. Yeah, I think we need a deeper understanding. I don't doubt that those machines make microgravity, but not everything which we associate to microgravity, like diffusion only, is maybe given. Yeah? We have to learn about limitations and about opportunities. Yeah.
No, thank you, Volker. And I think you've touched on to a very good point. It's about understanding what are the limitations with each associated technologies, and what are they ideally suitable for, and how we can maximize that. I think globally we still lack a little bit on the standardization of how we approach certain investigation and research. So I think that's very important. I'd just like to ask everyone, obviously everyone on the news, everywhere, everyone says talking about biomanufacturing, 3D printing parts and food and whatnot in space. With so much interest, what do you see as why hasn't there been more people getting involved in this area of research? Obviously, put funding aside, that's one thing. But why do you think? What are some of the limitations or challenges that is stopping or preventing people from wanting to do more microgravity research? So maybe I'll come back to Julia first. From a cell biology point of view, why isn't more people, especially in mechanobiology, using this unique environment to conduct more research?
Well, the reason why is the potential, I think, in translational medicine because I think it's really clear how space biology can build new knowledge to support human habitation in space, but it's not so clear how actually this can help in increase our quality of life. And that's the key point, because it's not like sending patient in space, but it's actually bringing the discovery here on Earth. And let's just think about the effect that the space environment has demonstrated to have on human physiology. And we have learned so much during this conference about, for example, cardiovascular deconditioning, or loss in bone and muscle density, or even vision impairments. And these are very interesting, all correlated. These biological effects are very close to aging and disease processes experienced here on Earth. And it's so evident, the connection, that I think going into deep in understanding this can really help to find other new solutions here on Earth on this disease. And I think the reason why, to go more in deep to your question, is that the human body has evolved like all other living organisms, has been evolving under the mechanical loaded condition that we have here on Earth, that basically is the persistent, inevitable constraint of the gravitational force field. And this is true in general, with the mechanical forces being the major regulators for physiological and pathological conditions starting at the cellular level. And so the concept of mechanical unloaded, the one encounter in space, which again is the reduction of the gravitational force, of course also affects cell function and overall tissue homeostasis, because in space, cell will no longer experience the constraint, the persistent constraint, but will actually have more degree of freedom, and this can lead the development of new molecular mechanisms. So in this sense, this is the nexus basically of space mechanobiology, which again is the study of how cells are influenced by this mechanical unloaded environment. And it's the area of space biology that mostly have the impact on translational medicine, because this will allow us to elucidate, to better understand molecular mechanism driving severe pathology. So the benefit is not just understand what happened to the human body when it's up there and so allowed for long-term space flight or exploration, but it's also try to uncover what is difficult to find on Earth because of the persistent gravity field, because these molecular mechanisms can actually not be discovered in normal gravity condition. And the first benefit that I think about, but of course, because it's my field of investigation, is cancer. Like cancer have been demonstrated to be highly sensitive to this mechanical environment, unloaded environment, by changing molecular mechanisms driving cell survival, malignancy, and other chemioresistance, for example. And this is just a benefit that we can get, like bringing discovery from space to Earth. So in this sense, space biology can really accelerate the discovery of new molecular targets for the development, let's say, next generation of mechanotherapies, molecular mechanotherapies. And this is really, really interesting.
Oh, thank you, Julia. And I totally agree with you. There's so much potentials. And I think now that we are developing more technology to support this type and advance this type of research, hopefully more researchers will get involved. Thank you. I'm going to ask Melanie. I mean, radiation is obviously not something people can just set up in their lab, so that's obviously a limitation in itself. But do you have any, if you were to suggest someone to advise someone on how to get started in space radiation, what would you say to them?
Yeah, I think I'm going to go a tiny different way here. I think there's no doubt for anyone here that it's useful research that will have consequences on life on Earth. I think there's maybe a tiny communication problem that could be that we could work on, which is that a lot of people see only the space and the going to Moon or Mars, and maybe we should just spread the word that we're not doing just for the sake of going to Mars or Moon, that it's useful technology then. And I think it's just, yeah, I think I'm gonna go for communication, and it's our job to all of us to spread the word and talk about that, and go to uni and give classes and courses to the new students to show them the potential of that type of research.
No, thank you. I think that's really important because I myself.
I mean, that's what you're doing with that conference, so that's good on you.
I think sometimes it really comes down to something as simple as communication and education, because I think most people have this conception that it is about sending and helping people exploring space, but more importantly, it's that duality and that translation down here on Earth. So no, I appreciate you raising a very good point.
No, it's very similar with radiation. A lot of people just see all the bad sides of radiation, so there's a lot of communication to be made on these subjects.
Yeah, no, absolutely, and hopefully we can slowly get that message across. Okay, thank you, Melanie. And over to Siddharth. I'm very curious as to you mentioned you're developing these new technology to support biological or pharmaceutical type of research. What do you see in the next 5-10 years, especially now with privatization of space, automations happening more people going into space? Where do you think, what would the next 5 years look like for space biology or pharmaceutical research, and how do we get people excited about this?
I think as more and more opportunities prop up and more events like the one that you're very, very nicely organizing, more these kind of events keep happening all around the world, we will finally break ice with the pharma and the biotech industry. I think that's one thing that is really, really important, to be able to self-sustain, because government funding will only go so far. If we really want to do breakthrough research, we really need the larger companies and corporates who are working in these areas to kind of support research. Because it's not, I think if you compare the amount of funding that's on microgravity research right now with the kind of funding that traditionally happens, it's like nothing, insignificant, right? So I agree with you. More FaceTime and communication, awareness, spreading awareness, and kind of highlighting the low-hanging fruits, like what are the things that we can achieve within the next 5 to 6 years. Because what tends to happen with space is that people are kind of, we try to fascinate people by showing them things that will not be happening in the next 20 years. So that's all good to attract people in. But when it comes to actually, the ones who actually are interested to do stuff, they think that we haven't reached that point yet. So there's no point talking about it yet. And that's where, things break. But we really should focus on bringing that out, and also collaborations. I think we have reached a point where there's labs across the world who can utilize any launch opportunity and a flight opportunity and fly their samples and get them back and have them expedited and brought back to them as well. So all of those kind of logistics of handling samples from space, bringing them back, getting companies, the conventional large biotech pharma companies to kind of come forward and support and kind of have like some kind of a roadmap in which they're kind of supporting the near-term goals, and then kind of having an image or a kind of a vision for outer space kind of stuff, right? So that's something I think that we need to collectively work towards, because every region has its own capabilities, but it's also challenges. So we have to kind of identify those, and once we have a fair idea, we might be utilizing Australian researchers using Indian flight opportunities, or vice versa, depending on how things go. You might even be able to launch experiments from Australia. Things are already looking up so well there. So there's so many things to kind of talk about, and I think the next 5 years, I think, to just summarize, more number of players coming in to provide these opportunities, both either suborbital or actual low Earth orbit flights, and also more interactions with pharma and biotech companies. And I think that will really open up what we really hope should have opened up by now. I think that should be my answer to your question.
No, thank you so much, and you've touched on quite a lot of key points. It's, obviously, pharma is a big one, and I think it comes back to what Melanie was saying. Is again coming down to simple communication and education on what are the values, and hopefully we'll be able to do that. And I really particularly like your statement about the global community coming together. Everyone has their strength, and also their technical abilities. So if we all come together to address these problems, I think that we will solve a lot of the challenges and issues for humans, both in space and down here on Earth. And you're very right. We like to sell the grand vision of people kicking footballs on Mars or dancing on the moon, but we have to be able to support that vision. So I think it's really great. Everyone here is making a contribution towards realizing that dream. So thank you very much. And finally, Volker, you're doing so many exciting things. Where do you see next 5-10 years, all the exciting things that will be happening? And where will we be?
Yeah, when looking back, that microreactors where I was involved took 15-20 years to come to attention, also in the academia. It may sound odd, but I would say it simply takes time. We have to accept that. And therefore, all these big new technologies, there is a valley of death where they go through. Microfluidics were almost dead in 2002 and then made a big home run. Yeah, some technologies really died. And why is that? It was said correctly. They are not correctly communicated. There was a time when you asked what is a microreactor. From 100 people, you got 100 different answers. And some said it's for big chemical production, some said for narrow. With such things, you kill industrial interest. Yeah? And then there's a tendency, pharma industry is really among the most innovative of all of them. But there's a tendency that everybody wants to be the follower, the second. Nobody wants to be the first who burns all the money and shows the other the way. So for all this reason, it needs a bit patience and time. But what we can work on is to have a similar narrative. I'm not saying we all have to say the same, that is against science, yeah. But our organization and flow chemistry broke through when Pfizer, Glaxo, Eli Lilly, and all those giants sat at one table, the so-called ACS Pharmaceutical Roundtable in 2007, and they said, behind any competition, we work now together on the technology. And we have to manage that they do this again for the pharmaceutical manufacturing in space. Yeah.
No, thank you so much. I totally agree with you. You know, I think, you know, I guess, you know, looking at the time, I think in summary, what everyone is echoing is one, communication, and two, it's not that there is no opportunity. The opportunities are all there. I think in some way, all the technology is also here to support at least the entry level into this area of research. I think it's about, you know, communicating, again, coming down to communication with the wider community on, you know, what are the values of it and why they should be excited. And I do agree with Volker that, you know, these things do take time. And I think, you know, even with just the satellites industry, it's taking all this time to really for everyone to see that value. So, but I think I'm very hopeful that, you know, humans, as more humans go into space, you know, having that experience, it is about the human experience. You know, more people will be more fascinated about, you know, what happens to the human body. And I think we live in a very exciting time where complementing technologies can help accelerate that kind of research. You know, data, you know, health monitoring, and so on. So it is a very exciting time to be involved in space biology and space health. Okay, and I'd like to thank all our panelists for their time and contribution and sharing their knowledge and expertise and experience. And I'd like to thank all our attendees wherever and whenever you're watching this. Hope we're able to inspire you. There is still a lot of challenges that we need to address. So, you know, hopefully we're able to inspire more people to get involved in all areas of space biology and space health research. Thank you again for your time, and we'll hopefully see you again next year. Thank you, everyone.
Thank you, Joshua. Bye.
Thank you, Joshua.
Thanks. That was great.
Thank you.
Bye.
Thank you very much.